She is professor of physiology at the oncology faculty of Ribeirão Preto, University of São Paulo, and a researcher at CMC. Well, Professor Elaine, she has so many collaborations in the world with different researchers from Mexico, Germany, France, Italy, and Brazil as well. And the main projects that underway in the laboratory of Professor Elaine are studying neurodegenerative, neuropsychiatric disease, experimental Parkinson's, analysis of neuroinflammation, in levels of linguistic kinesia, experimental schizophrenia as well, in which they study improvement of the nervous and the endocannabinoid system and sensory-motor function. She has received several national and international awards. In 2010, she was identified as one of the 50 women protagonists of Science Brazil in the area of the university science. She has been a member of the Latin American Academy of Science since 2020. She is part also of the Arctic School, and she was president of the Federation of Latin American Cardiology Society for Science and Neurotoxicity Society. So it's a great pleasure to be here, Professor London. So probably I will be presenting this of other speakers as well now. So the second speaker is you can yes, Professor Dr. Gisele Patroni. She is professor at the Department of Pharmacology and Technology Faculty of Pharmacy of the Federal University of Rio de Janeiro. She is master in pharmacology in the Federal University of Santa Catarina and a postdoctoral degree in neuroscience in the University of California, Miami. She has experience in the field of pharmacology and science, working mainly on the following topics: natural produced inflammation, immunotherapy, and neurodegenerative disease. And the next speaker is Professor José. She is professor at the University of Extremo Sul Catarinense, UNES. She has been a scientific leadership fellow since 2016. She is master in neuroscience and PhD by chemistry in the Federal University of Santa Catarina. She did a PhD in research at Universidad Autónoma de Madrid, Spain. Also, she did a postdoctorate in neuroscience laboratory at UNES. She works as a research neurodegenerative disease research line, and she was the recipient of the L'Oréal Nestlé Award for Women in Science in 2019. So welcome. And of course, the last one I will introduce myself a little fun, but just to let you know about what I have done. So I'm professor at the pharmacology department at Federal University of Paraná. I did my master and PhD doctorate in pharmacology at the Faculty of Medicine of Ribeirão Preto, University of São Paulo. I did two postdoctoral studies, one at Institute of Preto and another at National Institute of Drug Abuse in Baltimore, Maryland. And I have experience in the fields of pharmacology and neuroscience, working with following topics and psychopathologies associated with diabetes mellitus, pathophysiology and treatment, in addition to the neurobiology of side associated memory. So after this short presentation, we can start with Professor Learning now. Can I share? Can I share? Yeah. Explain. Can you see my screen?
Yeah.
Yes. Okay. So thank you very much, Janaína, for organizing this beautiful meeting. honor for me to be here with you all. It's a real pleasure. Hope next time it's going to be in Florianopolis, but all Ribeirão Preto. And I would like to thank you, Brazilian pharmacology, pharmacology society, because they accept our. So it's a great honor to be here between friends. Hope we can have a discussion at the end of my presentation or of the presentation. Okay, so I try to be provocative. So two birds, one stone. The neuroprotective effect of dog cycling on Parkinson's disease and Alzheimer's dyskinesia. What is two birds? What does it mean? One stone. Succeed in achieving two things with a single action. Let's see the prototype, okay? Okay, so I'm not talking a lot about Parkinson's disease, but it is a neurodegenerative disease. It was, there is no reason to believe that it's a kind of disease that mostly happens after the person is 60 years old, but nowadays, young age people also have it. Maybe it's not so, it can be mild, but there is this, and nobody knows exactly the cause. Because if they know, they could provide medicine for them. Okay? So, what happens in Parkinson's disease brain? There is a degradation of the dopamine substance. And what happens are those things. Okay? So it's difficult for the patient to walk, move, and to do several movements, several movements. But in 1968, after Eldon using three parts of patient, there was like a miracle. It's called honeymoon. All the patients have again movement and they feel very well. But during the last year, it appeared that chronic treatment with the L-alpha might induce abnormal movement and invoke those movements called the L-alpha brain. The cause of why it happens is difficult to say, and that's it. So what can I say that L-alpha gold standard for acute Parkinson's disease, but anyway, it's necessary to take drugs, neuroprotective, and all disease modifying action. So we're to prevent the progression of disease and reach side effects. It's difficult. There is a lot of research all over the world trying to do that. Okay, but in this presentation, I will talk a little bit about algorithm brain. What is the why people have side effects? It's a number, a lot of things. But today I'm going to discuss you about inflammation. But oxidative stress, protein activation, many reasons why a person, a Parkinson's patient might have. Okay, so okay, good. It's important before I talk about inflammation, dyskinesia and inflammation, to show that in the brain of Parkinson's disease patients, you can see here, it's a ligand, no? That can, and it shows many regions in the part of the patient brain that has a lot of microbeads that is the lesion. Okay? So if the brain stem, the basal ganglia, and some at the frontal cortex. Below, you can see the control brains, and the reason this kind of microglia detection. So, I'm not telling that all patients that have dyskinesia have inflammation, but for sure there is an increase in the inflammatory factors in the brain of the patients. Okay, but we have several evidence, no preclinical evidence in mice and rats that there is neuroinflammation in the brain of animals with L-induced dyskinesia. The first approach that we use was the NOS inhibitor. We know that nitric oxide is a messenger, a neurotransmitter, and it is related with part with inflammation. You can see here at the striatum or dorsal striatum of the brain of animals with dyskinesia, an increase, a lot of astrocytes labeled with GFAP protein. Okay, and those astrocytes they have the appearance to be activated, as you can see here. Uh, so L-DOPA induced dyskinesia increased the number of astrocytes, and the pre-treatment with 7-nitroindazole, the inhibitor of nitric oxide synthase, decreased dyskinesia and decrease the number of astrocytes, as you can see here. And those astrocytes in the dorsal striatum doesn't have the appearance of an activated state. Okay, but not just astrocytes. You can see here, it's dorsal striatum also, that there is an increase of INOS immunoreactivity, and those we didn't did the double labeling, but for sure it is glial cells. The glial cells are labeled with the inducible nitric oxide synthase. And you can see an increase in L-DOPA animals with L-DOPA dyskinesia and a decrease in the dorsal striatum of animals that have a decrease in dyskinesia and a decrease in treatment with L-DOPA. The same thing with the OX42 immunoreactivity that labels microglia, an increase in the dorsal striatum of animals who presenting L-DOPA induced dyskinesia, and when the dyskinesia is inhibited by 7-nitroindazole, we can see a decrease of in two regions, globus pallidus also, of the immunoreactivity for microglia. So we have at least two evidences of three presence of some inflammation in the central nervous system in the dorsal striatum. That was really beautiful because it was almost an accident, but the COX-2, the cyclooxygenase 2, is an enzyme that is constitutive in the brain, in the hippocampus and cortex, but not in the striatum, not. So after, in the brain of the animals with dyskinesia, as you can see here, there is an increase of the presence of COX-2 in the dorsal striatum, in neurons. We did the double labeling. And so COX-2 was induced in dyskinesia, in the striatum. And when we give the 7-nitroindazole, the animal didn't have dyskinesia, and the expression of FOX2 decrease. So several evidence that there is an increase of elements related to inflammation in the brain, in the striatum of animals presenting L-DOPA-induced dyskinesia. Okay, but what does it mean when I say two birds, one stone? I would like to talk with you a little bit about rosacea. What is rosacea? It is something that happens in the skin. It's a problem in the skin, okay? And it's very, here is represented by the red thing in the face of the designer. But doxycycline is really constantly used to treat rosacea, not in the antibiotic concentration, it's much smaller, the concentration. And in the Denmark, where they have a lot of real registration of treatment of patients and so on, they did an association, they observed one association of rosacea with Parkinson's disease. The patients that have rosacea have high incidence of Parkinson's disease than patients that doesn't have rosacea. And what was important, the patients that was receiving doxycycline treatment, there was a decrease in the Parkinson's disease incidence in those patients. That is interesting, no? And it goes together with our results. Just to show you some hypotheses of why rosacea can induce, this association with Parkinson's, Alzheimer, and migraine. It is a skin infection, and for sure, with this infection, some cytokines and other and some receptors are activated, and through the sorry, the nerve, the trigeminal nerve, it goes in some way, goes to the central nervous system. And there is an increase in microglia and astrocytes in some regions of the brain, and maybe an association between rosacea and those diseases. It is an hypothesis and has been tested in many laboratories. But we have another nice history. This is what the medical doctors call anecdotal evidence. When we published the paper in 2016, this lady, her name is Rosie Brown, she's an English nurse, and she was for many, many years in Uganda. And doxycycline can be used to prevent malaria. So what happened? Rosie was diagnosed in 2015 with Parkinson's disease, and she wrote to us in 2000, and she has been taking doxycycline all the time. She was in Uganda, no? 10 milligrams each day. And what happened? The symptoms of Parkinson did not evolve. So she was well for all the time, more than 5 or 6 years with the doxy treatment. She went to UK, she went back now in 2018, and she continues to use the doxycycline. But nowadays, she had some problems. She just wrote to me, and she's stopping taking doxy. We hope her symptoms won't get worse. So it's an anecdotal evidence, but it's very interesting, and we receive information from many others, okay? But the first of the two birds, we, by accident, doesn't matter, I'm not telling you the whole history. If you want, we can talk about it later. Some animals that were received the microinjection of 6-hydroxydopamine that is used to produce dopaminergic immune lesion, they receive food that had doxycycline. It was so interesting, such an incredible surprise that those animals, it was 38 mice, they didn't develop the lesion. As you can see here, those are controls. Here we have the lesion in the example. This is tyrosine hydroxylase immunocytochemistry. And when the animal receives doxycycline in the food or either IP injection, because subcutaneous, I'm sorry, because we want to prove that it was related with doxycycline, or it means not just in the food, injecting. And it was incredible. The animal didn't develop the lesion. So our results, the evidence that we got, it goes together with the described in literature for men and our anecdotal evidence. But there was another one. This is more interesting. A doctor, a young doctor in the U.S., he works with patients with Parkinson's disease, and some of their best patients they have severe freezing of gait. They couldn't walk. Okay, and those patients have sinusitis, and he needs to give doxycycline for the patient, and he described the thing as a miracle because the patient started to walk and they were very well during the time that they received doxycycline. So it means another anecdotal evidence that doxycycline can be can be used in humans. Okay, so now we have this beautiful hypothesis together with the information we had before with 7-nitroindazole, that there is an inflammatory reaction in the brain of, in this case, animals that have L-DOPA-induced dyskinesia, in the human beings also, and maybe doxycycline can be used to treat dyskinesia. I know that is a slide full of things, but what I want to show you and it is important, is here. Sorry. You can see that the animals, those are the sum of abnormal involuntary movements 1, 7, and 14 days after receiving L-DOPA. And when they receive doxycycline, what happens is that the dyskinesia went down. And you can see here also, those are the animals that receive doxycycline. And the most important thing is that the doxycycline was active against the dyskinesia, but didn't disturb any of the movements, the improvement that L-DOPA provided to patients. Okay, we saw also that at the animals with dyskinesia, there was also an increase in COX-2, astrocytes, and microglia, and doxycycline decreasing the L-DOPA-induced dyskinesia also decreased the expression of COX-2 activated astrocytes and microglia. So a similar effect that we have showed before for the nitric oxide. Another very nice thing you can see here. Here you have the animals with the dyskinesia that is increasing during the time of that the animals receive L-DOPA. And if doxycycline is given to the animals since the first day, they didn't develop dyskinesia. Okay, and in this publication, we were able, I'm sorry, we need to go back. we were able to show that in L-DOPA-induced dyskinesia, there was an increase of ROS that was decreased by DOX, and also an increase of metalloproteinase 9 and 2. And this was decreased also by DOX when the dyskinesia was off. So two more demonstrations, two more evidence that doxy was important. You can see here that dyskinesia was associated with increase of cytokines. This is PGE2, here TNF alpha, and you can see that doxy decrease the induction of both cytokines. Okay, I don't know my time. It is important to say that doxy it's much more than an antibiotic. Okay. Now we, in association with my friends in Paris and in Argentina, we were able to show that the doxy can decrease the alpha-synuclein aggregation. The alpha-synuclein can constitute the corpus of Levy, and that is a real sign of a real evidence of Parkinson's disease. So, and we did many more things that is not the time to say now. Okay, everything was published, and now we are trying to put everything together, and as you can see, the treatment of doxy in animals that present L-DOPA-induced dyskinesia or Parkinson's disease, we have evidence of neuroprotection concerning the TH, tyrosine hydroxylase immunocytochemistry and behavior. We have a decrease of microglia and astrocytes. Also, we are doing some studies with the NG2, that is the fourth type of glia. We have an increase in the oxidative stress, ROS. We saw neuroinflammation, metalloproteinase, and COX-2, and alpha-synuclein aggregation. So maybe doxy can be used for treatment in patients and animals. And if I have just 1 minute, just to say that with the people in France, we have been working. They are synthesizing molecules of doxy that doesn't have antibiotic activity. And so we are going to test those drugs in the preclinical experiment. So I want to say thank you very much again to Janaína, again to the Society of Pharmacology, and that is something that is in my heart. And I'm ready for questions, wherever you wish, Janaína. Thank you very much.
Thanks, Professor Elaine. Wow, that's so beautiful data. And I think this kind of question, like the positioning of medicine is...
Não te escuto, Janaína. Não? Seu microfone.
Era para estar funcionando.
Eu escuto.
Eu também escuto.
O que será que aconteceu então? Mas... ok. Professor Elaine? Não?
Não.
Ibrahim, por favor, então, leia a questão. Nós temos 5 minutos. We have 5 minutes to question.
O ideal é que ela entre e saia.
As pessoas estão ouvindo, mas
você me ouve?
Não estou.
O problema é com o computador dela. Manda mensagem para ela entrar e sair. Carol, você pode nos ajudar?
Assim.
Eu estou te ouvindo agora. O problema era esse.
Ah, então perfeito.
Desculpem.
No, we have 5 minutes, so there are some questions. I have write some questions, and Felipe Porto also, if you can read and try to answer. We have 5 minutes. We can do it now.
I can answer the first question. I would love to do this investigation with you, no?
Yeah.
The effect of doxy in animal model of diabetes and others involving encephalopathy. It's going to be a pleasure. Now we have, we know well the drug, and Felipe Porto, could doxy be acting as an anti-aging impairing cell senescence, as it could reduce ROS and pro-inflammatory cytokines? Maybe yes. We don't know. We didn't investigate that. It's a good question, and we have been preparing ourselves to do the Parkinson's, to induce the Parkinson's in older animals. So we need to think about very well how to design this kind of experiments. But what can I tell you is that I never, and I read a lot about doxy effects. I never saw any description or any study about doxy effect in senescence. Okay, but would be nice if to make a more deep source in the literature. Thank you for your question, Philippe.
Thank you, Professor Laine. So we could discuss a lot because it's very, very interesting, your data, your findings, and I think the inflammation, oxidative stress is a key factor between all the relations. For example, Parkinson's disease, the severity is higher in diabetic patients, so there is this kind of discussion. So probably some drug like oxy could be very, very interesting. So thank you a lot, Professor Laine. If someone have other questions, please write in chat, and if we have time at the end, we could also discuss more about these data. So, but now I invite the second speaker, Professor Gisele. So please. I will try to take care of the time, so I can send some message private for you just to let you know about your time. If you don't need to worry.
Okay, thank you, Janaína. Congratulations, Elaine. It's a beautiful talk. I have a lot of questions that we can discuss a bit more in the end, so we don't get too late. Let me share my. Can you see my slide?
Yes.
Okay. So now we go from Parkinson's to viral infections. I want to start by thanking the organizing committee and Janaína for inviting us to this all-women session. It is a great honor. And what I brought today is some of more very exciting data on how cognitive dysfunction following viral infection can be mediated by neuroinflammation. So we do believe that neuroinflammation could be a therapeutic target. So Brazil has been facing different emerging infectious diseases in the past years, especially the ones caused by arboviruses, which are transmitted by mosquitoes, especially mosquitoes from Aedes canis. So we've been having outbreaks of dengue virus and Zika virus and chikungunya virus, and more recently, like the rest of the world, we face SARS-CoV-2, the pandemic. Our lab has been working with Zika virus and chikungunya virus for quite a few years, I think since 2016. And we are now interested in SARS-CoV-2 as well. So today I'm going to show data on chikungunya virus and SARS-CoV-2, which are our most recent data. So chikungunya is, as I said, it's transmitted by mosquito bites, and it was first described in 1952 in Tanzania, and ever since then it has been causing outbreaks throughout the world. We have the affected areas marked in purple here. And in Brazil, we had two important outbreaks, the first one in 2004 and the second one in 2017. And with the end of the SARS-CoV-2 pandemic, we are seeing a new surge in new cases as people are leaving home again. So we are the Ministry of Health is expecting the numbers to go up again soon. So chikungunya virus causes disease that is usually self-limited. The patient has fever and it has muscle pain and joint pain. But in some patients, we see the virus inducing a arthritis-like alterations. So in some people, those alterations, they may last for months or even years. And chikungunya in the Tanzanian language, it means the one who bends because of those joint alterations. But what called our attention is that in the recent outbreak, there is an increase in the number of people that present injuries in other organs, not only the joints, especially neurological alterations like encephalitis and neurocognitive impairment. So our PhD student, Susan Araújo, is interested in studying how the virus impacts the central nervous system, and she's looking for the mechanisms underlying this event. So to study the effect of the virus on the brain, she established a model where she injects the virus directly in the brain. Actually, it's an intracerebroventricular infection. And when she infects male police mice, adult mice, she found that the virus could replicate in the brain. So here we have viral RNA, and this replication is time dependent. So we see a peak in the replication between 3 and 6 days post-infection. And what we do believe is that the virus is replicating in specific cells. So we first looked at whether the virus was replicating in microglial cells. So we use J2 that recognizes viral material, and we try to see if it's localized with the IBO1 markers for microglia, and we saw no localization. We also did not see localization with astrocytes. Here we use the GFAP marker. And what we do believe is that this replication is happening in neuronal cells, although we still have to prove it. And when we look at the brain structure, what called our attention was that viral replication was happening especially in the cortex and hippocampus, that are areas that are important for cognition. So to see if this replication could mean behavioral alterations, we used a very simple memory test, the object recognition test. In the test, the mouse is exposed to 2 objects that are similar, and in the test session, we change one of the objects with a new one. So The control mice, they remember the familiar object, so they spend most of the time exploring the new object. However, when we evaluate the infected mice after 7 days, those mice could not recognize the familiar object. So they spend half time exploring each object. So we did have cognitive impairment in models. Next, we wanted to see whether this behavioral alteration was connected to any synaptic dysfunction in the hippocampus of these mice. So we used two markers. We used an antibody against synaptophysin to stain for presynaptic terminals, and HOMER1 to stain for postsynaptic terminals. And when we did the confocal microscopy, what we saw was that control animals, they have the yellow points that you see here that are points of localization of the two markers, meaning that this represents the synaptic function. And when we looked at the brain of the animals infected 7 days before, we saw a significant decrease in the density of this synaptic function. So mice had behavioral alterations that were associated to synaptic loss. We then looked at whether the synaptic loss could be due to a neuroinflammation. Then we used IBA1 again to stain for microglia and looked at the hippocampus of these mice, and we checked three different areas: T1, T3, and also dentate gyrus. And what we saw was that in all three areas we had an increase in the number of IBA1 positive cells compared to the control mice. And when we performed a morphological analysis of this microglia, what we saw was that the infected mice had significantly increased levels of type 4 and type 5 microglia, which are considered reactive phenotypes, as you can see here in the picture. So we knew that our microglia was activated, and we confirmed that also those in the level of pro-inflammatory markers. So we did find an increase in the messenger RNA for interferon beta and also for its receptor, which was expected because the organism mounts interferon type 1 response when it's challenged with a virus. And we also found increased levels of TNF alpha and complement protein Q1Q. This was very exciting for us because we knew from this beautiful paper from Stevens published in 2016, we knew that complement activation and microglia, they mediate synaptic loss in Alzheimer's disease, which is a disease where we also have memory impairment. And this process is called synaptic pruning, and we thought that this would be happening after chikungunya infection. So synaptic pruning it happens when you have C1q deposition in synaptic terminals. The C1q is going to induce the conversion of C3 in C3b, and the C3b is recognized by complement receptors that are expressed by activated microglia. So this works as a eat-me signal. Then the microglia just goes there and eats the synapse, the phagocytes, the synapse. This is very important in a physiological context because when you have, when the brain is developing, it has a lot of excess synaptic function, and the body needs to get rid of this excess synapses. But when it happens in the mature brain, what you have is that you are eating important synapses, and with that you have synaptic dysfunction and memory impairment. So we wanted to see if this was happening in our model. So we knew that we had increased C1q levels, And then we used an approach, the same approach that Beck Stevens used, that was to perform co-localization studies using synaptophysin for the synaptic terminal and IVA1 for microglia. And then you can see that the microglia, it co-localizes with the terminal, showing that this microglia might be eating those synaptic terminals. So with that, we show that neuroinflammation and synaptic pruning are the mechanisms underlying cognitive dysfunction after chikungunya virus infection. Well, we were advancing in this project, but then, like everyone else, we had to shut down our labs and go home because of COVID-19. So as we were home, trying to write reviews and reading, we started seeing emerging evidence of clinical, of neurological manifestations. So the patients, they were having not only lung alterations, but also alterations in central and peripheral nervous system. So a lot of people were presenting loss of smell and taste, and we were seeing a lot of cases of encephalopathy, encephalitis, cerebral vascular disease. But what really called our attention was the emergence of cognitive dysfunction cases. Even in mild or asymptomatic cases, we were seeing cognitive dysfunction. And we were very interested in looking at the mechanisms underlying this phenomenon. The thing is, we did not have access to an NV3 facility to infect the mice. So at first we could not work with the virus. But then evidence started to emerge that some viral proteins could actually cross the blood-brain barrier in both mice and in human in vitro models, more specifically, the SARS-CoV spike protein. That is the protein that covers the viral surface, and is the protein that the virus uses to infect the host cell by binding to ACE2 receptors. At that time, Professor Leda Castillo from UFRJ, she was synthesizing spike protein to develop ELISA kits for diagnosis, and she was kind enough to give us some spike protein for us to inject our mice. And then, she was our postdoc at the time, Fabricia Dantas. She's now a professor at the Rio State University. So she developed the model where she was injecting spike protein directly in the brain, also through intracerebral ventricular route. And then after 6 days, she exposed mice to the object recognition test if they had memory impairment. So at 6 days, we saw nothing. The spike injected mice, they could recognize the familiar object. And then we looked again at 2 weeks. Again, nothing happened. And we were almost giving up the project when we decided to give one last try at 30 days. And then at 30 days, spike-treated mice could no longer recognize the familiar object. So then we looked again at 45 days, and still the cognitive impairment was persistent. And we got really, really excited because this was very similar to what was happening in people with mild COVID. So the same late cognitive impairment that we were seeing in people was also happening in our mice. So we next rushed to see how the synapses of those mice were. And when we looked at the brain in 7 days, we saw no changes in the number of synapses in the hippocampus. But at 45 days, we saw a significant decrease in the localization of the synaptic concept. Then we imagined that the synaptic loss would be associated with neuroinflammation. That was our hope. Then we next went to look at how the microglia of these mice work. At 7 days, there was no increase in the number of IBO1 positive cells, nor changes in levels of inflammatory markers. But at 45 days, we saw an increase in activated microglia in the hippocampus of the spike-treated mice, and we also saw increased levels of CNS alpha, IL-1 beta, and also type 1 interferon. So we thought, okay, there is microglial activation, there is new inflammation happening. Do we have synaptic running? So we measured C1q mRNA in those brains. In the early phase, no changes, but at 45 days, there was significant increased levels of C1q in those hippocampus. And then we looked at 2 hippocampal regions to confirm the increased colocalization of the microglia with the synapses. So we did have synaptic pruning at the late phase of our model. And then to validate the role of C1q in the cognitive dysfunction in the spike, we blocked C1q action using a specific C1q antibody. So we treated the animal for 30 days, and after 30 days, we performed the object recognition test. And so when we block C1q action, we can protect mice from the cognitive impairment in this biospike. But we still didn't know how spike was acting. We knew that it could not be through H2 receptors. It was not the same receptor that the virus uses to enter the cell, because mice, mouse 2, H2, have a very low binding affinity for spike protein when compared to humans or monkeys. Then we started to think that it could be a pattern recognition receptor. We first thought of toll-like receptor 7, since we're talking about the single-strand RNA virus, and we performed some preliminary studies, but we could not find any good results here. And then those beautiful studies came out showing that both in silico and in vitro, that spike2 could interact with toll-like receptor 4 and activate it. And well, we didn't think of toll-like receptor 4 at the beginning, because toll-like receptor 4 recognizes enterotoxin, so gram-negative bacteria. But we did have increased levels of toll-like receptor 4 mRNA in our model, just only in the late phase. So we got really excited. And to check if toll-like receptor activation was the mechanism underlying spike effect, we blocked its activation with the farmacological inhibitor TAC242. So we treated the mice, and then on the 30th day, we performed the object recognition test. So TAC242 protected the mice from spike-induced behavioral alterations. And we also confirmed that using toll-like receptor for knockout mice. So when we inject toll-like receptor for knockout mice with spike, we first looked at the behavior at 7 days, and they performed well. And then we were concerned, very interested in looking at the late time point of the model. So at 45 days, toll-like receptor 4 mice, they don't develop the cognitive impairment compared to wild type mice. So we looked at the hippocampus of these mice, and these mice had a lot, significantly increased number of synaptic functions compared to our wild type mice. So in the absence of toll-like receptor 4 signaling, we have less synaptic loss, and we have a prevention of the cognitive impairment. Those mice also had less activated microglia in their hippocampus and less synaptic pruning. So, I don't know how much time is.
21 minutes.
Oh, okay, I'm good. So I can show the last set of data. So we wanted to see whether our findings had translational relevance. So for that, we came up with Professor Juanita Leon. She's a neurologist at Rio, and she assessed a cohort of 86 individuals with confirmed COVID-19 diagnosis. And most of them were mild cases. A lot of them were asymptomatic. I believe only 6 of those patients needed to be hospitalized. And after 6 months after the diagnosis, she performed cognitive assessment in this patient. And almost 47% of those patients, they had post-COVID-19 cognitive impairment. So to see toll-like receptors could have any effect on that, we collected blood from these patients. We performed genotyping for a couple of toll-like receptor polymorphisms. And what we found was an interesting response for this specific polymorphism, the 260 G8. So people that carry the GG genotype for this polymorphism, they have a significant increased risk for cognitive impairment after COVID-19. So with that, we showed that both neuroinflammation and microglial-mediated synaptic pruning are the mechanisms that respond for cognitive dysfunction in both chikungunya and SARS-CoV-2 viral infections. And in the case of SARS-CoV-2, when we block toll-like receptor signaling, we can prevent those alterations. We showed that toll-like receptor polymorphisms, they define the cognitive outcome in humans. So we believe that toll-like receptor 4 is a potential target to prevent COVID-associated cognitive dysfunction. So I would like to end by thanking Fabricia and Susana, which are the first authors in studies that I showed, as well as my colleague Claudia Figueiredo, also from UFRJ, that is the last author in the studies together with me, as well as the other students in our lab that helped in those studies, and our collaborators from other labs at UFRJ. And to finish, I would like to thank our funding agency. Thank you.
Thank you, Gisele, for the brilliant presentation. Very nice data. It's curious. So we have 5 minutes. So there is one question. I have made this question. Just something that I am worried about it. It's the emotion, depression, anxiety. And what we have seen is, at least when I am looking for, I have seen this correlation to besides the cognitive impairment. So my question is, since the prefrontal cortex and hippocampus are also important to the emotional mediation, do you have an idea about the impact of these infections, SARS-CoV-2 and even chikungunya, on depressive behavior or depression in humans?
That is a very good question. We are seeing a new pandemic of depression right now. We both related to the disease and unrelated. People were confined for 2 years, and a lot of people are struggling to go back to normal life. We are also interested in looking at those alterations. We performed a few depression tests in our mice. So we did the tail extension test, and we have some interesting data on that as well. Yeah. So it seems like it could also be affecting, it could also be inducing depressive-like symptoms.
Yeah.
our model at least. So it is definitely something that we want to investigate deeper.
Yeah. So, okay. So thank you, Gisele. I really appreciate your accepting to participate in the symposium. So that's very, very nice. I didn't know in person you, but now we can talk a little bit more. It's very nice. So now I will invite the Professor Josiane to perform our presentation. And if someone has some questions, please don't be shy, write in the chat. Probably you have 5-10 minutes after in the end, so please.
Can you see my presentation?
Yes.
Yes, we can.
Okay. Good morning. Thank you for inviting Aina. It's a pleasure for me to stay here with you, share this important moment for science. Now I will talk about Alzheimer's disease and depression. What is the role of neuroinflammation? The pathogenic marks of Alzheimer's disease involve A beta plaques in tau neurofibrillary tangles. In early stage, 45 years old, the formation of a barrel core due to abnormal cleavage of amyloid precursor protein, APP, forming a beta monomer. A beta monomer are intrinsically disordered and have to propensity to oligomerize and aggregate into a beta plaque. A beta plaques active microglia and astrocytes. A beta also induce tau hypophosphorylation and subsequently form tau tangles and neurofibrillary tangles. Leads synaptic and neuronal loss, causing neurodegeneration. The focus of this presentation is neuroinflammation. Many studies show the link between AD and neuroinflammation, include inflammasome activation. How does this happen? Here I show you a beta species here can trigger toll-like and twin2. Toll-like is the main way to activate the inflammasome. Toll-like receptor induce activation of NF-kappa B that induce transcription of NLRP3 protein. This protein is an important compound of inflammation. Inflammation involves it's a complex protein of NLRP3 protein, an adapter protein S, caspase 1. This caspase 1 is responsible to mature interleukin, né? 1 beta, interleukin 18. Mature this interleukin and release to activate others microglial cells to amplify the neuroinflammation. Many risk factors are responsible for the development of aging, include genetic aging, bacteria, diabetes, gut dysbiosis, hypercholesterolemia, obesity, virus. These risk factors induce systemic inflammation and blood-brain barrier disruption, and in turn induce A-beta aggregation, tau hyperphosphorylation, and neuroinflammation. A-beta aggregation here and tau hyperphosphorylation. And neuroinflammation, glial neuroinflammation. The neuroinflammation involves release of inflammatory mediators like nitric oxide, interleukin 1 beta, interleukin 18, TNF alpha, prostaglandins, and chemokines like fractalkines and IP1 alpha, IP10, and CP1. Then these events lead to neuronal death, memory loss, changes causing memory loss, changes in mood and personality, lead to dementia like Alzheimer's disease. This, okay. In depression, what are physiopathological mechanisms? A comprehensive understanding of depression pathogenic should consider interaction between various systems and pathways. Here I showed you various pathological mechanisms like dysfunction of neurotransmitters, dysfunction of neurotrophic factors, stress and dysfunction of HPA axis, metabolic disorders, microbiome gut-brain axis, and neuroinflammation. This is presentation key. Here, this is a timeline. This timeline shows discovering and associating depression with immunological immunological and inflammatory changes. We observed that discovery of hyperactivation theory increased the cytokine productivity was 20,000. And since 20,000, many articles, many research and review articles were published about neuroinflammation. Multiple mechanisms may contribute to to the dysregulation of cytokines in depression. The HPA axis and sympathetic nervous systems are activated to various types of stress, both cortisol and catecholamines such as acetylcholine regulate inflammation here, acting as immunosuppressant, acting as an immunosuppressant, inhibition leukocyte trafficking, and activating inflammatory cytokine product, as seen here. Alarmins are produced in brain in response to stress and trigger toll-like receptor pathway. Just a minute to connect to my. Sorry. Continue. Sorry. Alarmins here are produced in the brain in response to stress and trigger toll-like receptor pathway depends cytokine production here. Alarmin activate toll-like receptor 4 and induce activation of NF-kappa B and induce production cytokine production. And this way, this pathway to activate inflammation too, that in turn produce interleukin 18, induce tissue damage. Other mechanisms involved in neuroinflammation is microbial compounds. Dysregulate peripheral immune response in depressive patients and may also result from changes in microbial level. The microbiome has increasingly been implicated in shaping the immune response in brain function. Alterate the cytokine or increase the cytokine production. This different way increase the cytokine production and causing tissue damage. So what is the role of neuroinflammation? Um, the both of both uh, pathological conditions, depression and Alzheimer's disease. Here, uh, uh, we show you the HPA axis dysfunction, chronic inflammation, and neurotrophic factors, and are central role in pathological of both depression and Alzheimer's disease. The increased levels of uh, glucocorticoids, increase of pro-inflammatory cytokine, and decrease the genetic and serotonin induce maybe lead increased vulnerability to a beta toxicity and hippocampal atrophy. Um, this does contribute the progression from depression to Alzheimer's disease. Alzheimer's disease. Here I show you the link uh, of early life programming with Alzheimer's disease. Evidence supports the role of early life program in sustainability for adult neurodegenerative disease like Alzheimer's disease. Here, the stress hormone, cytokines, bacteria, or viral are transferred through the maternal fetal circulation here. And then these factors induce or modulate changes in microglia in the fetal brain mediated by epigenetic changes. Here are the effectors like inflammation, prenatal stress, glial energy reserve, neurodegeneration, immune senescence, together will modulate the individual's phenotype. And then over a sequence of repetitive, potentially asymptomatic modification in 1, 2, 3, child or adult neural and glial development are shaped with variable degrees of susceptibility to neurodegeneration. And then here, genetic environment and stressors give rise to factors that contribute to depression in late life, including neurodegeneration, cerebrovascular disease, chronic inflammation, all of which can create a brain that increasingly vulnerable to the risk of development and progressing of cognitive decline due aging. Factors associated with resilience including social support and participation, physical and cognitive engagement, cognitive reserve, can buffer even vulnerable brains from susceptibility to the behavioral phenotype of aging. Here, I show quickly our results about one is performing in our laboratory. We performed this study to evaluate the neuroinflammation in an animal model of depression and dementia. The results of this study will submit it to neurobiology of aging. In this study, the animals were subjected to chronic mild stress protocol and after received a beta-142 peptide injection. 24 hours after the surgery, they were treated with fluoxetine, memantine, galantamine, donepezil in association of memantine, galantamine, or donepezil with fluoxetine. The results of this study indicated that two animal models, depression and dementia, increased interleukin-1 beta and TNF alpha levels in the brain. As in isolated models, thus not indicating a synergistic effect after the two models. I didn't show the results because of time. And then thank you all. Thank you all. PhD, master's degree, undergraduate student of my lab, collaborative researchers. Our research group on neurodegenerative disease of the experimental neurology laboratory thanks the financial support L'Oréal, Brasil, Unesco, Academia Brasileira de Ciências, in the Formula Insights 2019 award. We thank CAP, CNPq, and PAPEC. Thank you.
Thank you, Josiane, for your talk. Well, for me, it has been very clear the involvement of neuroinflammation in all these diseases. It's curious, so just to think better about it, you know. I have a question. Would you know something about the involvement of AMP kinase in this whole process involving Alzheimer's disease, depression, and also diabetes? I don't know if you read something or have did some experiments involving this pathway.
I didn't experiment yet about this, but the AMP kinase. I don't remember the principal way.
No. Yeah, because I was reading recently and I am interested in study a little bit more about AMP kinase because this pathway regulates autophagy, inflammation.
Yes.
Yeah.
The I think diabetes and Alzheimer's disease, like depression, involve autophagy, dysregulation of autophagy, and MPQ, MP activating protein kinase, okay. But in the uh, certainly point the connection I didn't understand yet.
Yeah, no problem. But thank you again for accepting
Thank
the
you.
invitation. So the last talk it should be performed by me. I did a video because I feel more comfortable. So let's see. Yes. Thank you, Ibrahim. The title of my talk is Impact of Type 1 Diabetes Mellitus on the Development of Depression. Would neuroinflammation be a key factor? Diabetes mellitus is the most prevalent metabolic disorder and is becoming a serious worldwide public health threat. It is estimated growth of around 46% from 2021 to 2045. When we look at different regions or countries, we can observe that this estimate is even higher. The hyperglycemia is the main feature of diabetes mellitus, and it's related to several complications like blindness, renal impairment, cardiovascular disease, neuropathy, deficit in learning and memory, anxiety, depression. Today, I will focus more on depression. The prevalence of depression reaches 30–40% between diabetic individuals when compared to non-diabetic population. However, only 2–3% of cases of depression between diabetic patients are diagnosed and treated correctly. This situation can further aggravate the diabetic condition and also the necessary care. Here, just to let you know that this review is very interesting because they discuss about emotional regulation and diabetes distress in adults with both types of diabetes. So this is a very interesting review highlighting that this topic has finally gained great interest in the scientific community. Several research have been proposed that there is a bidirectional relationship between diabetes and depression. So in this diagram, we can observe that hyperglycemia could be promote or facilitate the depression, and also depression may precipitate the diabetes or worsening the diabetic condition. The main treatment of the depression is with antidepressant therapy, and this therapy induces favorable effect on depression, but only in some percentage of patients. Moreover, this induces slow therapeutic effect, it worsens some other diabetes comorbidities, and also there are reports of interactions with other medications. there is a low adherence rate to the treatment, and some antidepressants present potential for impairing the glycemic control. So we need to better understand the pathophysiology of the depression associated with diabetes mellitus, as well as looking for a more effective treatments. It is known that hyperglycemia is able to induce functional and structural changes in the brain, leading to the encephalopathy and also to the dementia and other psychiatric disorders like depression. In our lab, we use it as experimental type 1 diabetes mellitus, one injection of streptozotocin. This compound recognizes the GLUT2 receptor that is more abundant on pancreatic beta cells, and it's able to induce a beta cell mass reduction, insulin insufficiency, and consequently chronic hyperglycemia. It's important to mention that streptozotocin is one of the most used models to induce experimental diabetes mellitus. In our lab and other groups also showed that these compounds, especially after 4 weeks of this diabetes induction, these animals present an increase in glycemia, an increase in glycated hemoglobin, and also a decrease in the weight gain of these animals, which reinforce the some diabetic condition parameters. When we evaluated the animals in the forces field test, what we observed is a more pronounced depressive-like behavior when compared to normal glycemic animals. For example, these diabetic animals present a decrease in the first latency to enter in immobility, and also an increase in immobility frequency and decrease in the active behaviors like swimming and climbing frequencies. In this study, the authors showed that type 1 diabetes aggravates post-hemorrhagic stroke cognitive impairment by augmenting oxidative stress and neuroinflammation in mice. Regarding to neuroinflammation, we observe that diabetic condition impairs this neuroinflammation, showing a higher microglial cell activation. We can observe here, for example, when they evaluate through fluorescence intensity. In the same way, the same authors, they also demonstrated a higher microglial cell count in these post-stroke animals plus diabetes, highlighting that the diabetic condition impairs this inflammation process. But how could neuroinflammation be a key factor in the development of depression associated with diabetes mellitus? So we performed a study to better understand the involvement of immune system activation with depression associated to diabetes mellitus. So we studied the indoleamine 2,3 deoxygenase kynurenine pathway, so we call IDO enzyme, as a potential pharmacological target to treat depression associated with diabetes. These several factors like psychological stress, stroke, Parkinson's disease, sclerosis, Alzheimer's disease, and other factors, including pro-inflammatory cytokines, are able to increase the activity of IDO. It's known that these enzymes leads to the lower plasma tryptophan and an increased synthesis of detrimental tryptophan catabolites, TRICATs, both of which contribute to the onset of depression. and consequently, there is a reduction in the serotonin synthesis. So here we can observe better the tryptophan is an essential amino acid that is precursor for serotonin synthesis. Once IDO is activated, for example, by pro-inflammatory cytokines, uh, this enzyme will be able to reduce the serotonin synthesis and increase the tryptophan catabolites like quinorenin and quinolinic acid, for example. Both of these catabolites are able to cause neurotoxic effects. For example, here it has been shown that these catabolites are able to induce depressogenic and anxiogenic effects. They activate oxidative pathways and increase neuroinflammation. They cause mitochondrial dysfunctions and have neuroexcitatory neurotoxic effects that may lead to the neurodegeneration. So in our study, we observed after 4 weeks of diabetes induction that these diabetic animals present in the hippocampus an increase in pro-inflammatory cytokines like TNF-alpha, interleukin-1 beta, interleukin-6. In this more recent study, Bambi and co-workers also showed this increase in TNF-alpha in the hippocampus of diabetic animals, but also in the prefrontal cortex. Curiously, they demonstrated a decrease in the anti-inflammatory cytokine interleukin-10 in the prefrontal cortex and also in the hippocampus of these diabetic animals, reinforcing this neuroinflammation process in these brain areas which are important in the emotion mediation. In our study, we observed an increase in the IDO expression in the hippocampus of these diabetic animals. And also we observed a decrease in the serotonin levels and also noradrenaline levels in the hippocampus of these animals, which seems to be in accordance with our hypothesis and with have been proposed to several researchers. In the same study, Bambi and co-workers observed also this increase in the IDO expression in the hippocampus, but also in prefrontal cortex, reinforcing our previous data. And also they demonstrated a more pronounced depressive-like behaviors, also corroborating our data, showing an increase in the mobility time when the animal were evaluated through tail suspension tests, and also an increase in mobility time when they were submitted to the forced swimming test. When we performed a prolonged treatment with minocycline, which is indirect inhibitor of IDO, what we observed was an antidepressant-like effect. So this treatment was able to decrease the mobility frequency and increase swimming frequency, and no change was observed in the climbing frequency. Here, it's important to mention that several have been shown a relation between increase in swimming with an increase in serotonin levels in some brain areas like hippocampus. So we do not measure serotonin levels, but we could suggest that probably this increase occur. When we treated these diabetic animals with a non-steroidal anti-inflammatory, the ibuprofen, we observed exactly the same, a decrease in the mobility frequency and an increase in the swimming frequency, but no changing in the climb. So also we that probably an increase in serotonin could be occur in this situation. So, although in this study we did not measure the level of serotonin in the hippocampus after the different treatments, the opposite also seems to be true because we treated the animals with the fluoxetine, which is an antidepressant drug able to increase the serotonin availability. So here in this study, we observed that the fluoxetine treatment was able to decrease immobility frequency and also increase swimming frequency, but no change was observed in the climbing frequency. So corroborating our hypothesis that serotonin could be, an increase in serotonin levels is involved with this antidepressant-like effect. Interesting, after all treatments, the expression of idle was reduced. So we did not measure it, which was a limitation of our study. We did not measure the idle activity, just the expression. But it's curious that the treatment was able to decrease, and we could infer probably the activity could be also decreased by all this treatment. It's a very nice overview of the effects of streptozotocin. So here we can observe the effects of hyperglycemia, increasing advanced glycation end products, and also these will activate the transcription factors like NF-kappa B. And in these situations, this transcriptional factor will increase the inflammatory cytokines, which are able to activate the idle activity, will lead to the increase in toxic metabolic production and also decrease in serotonin synthesis and release. And also the inflammatory cytokines are able to activate inducible nitric oxide synthesis synthetase enzyme, and this will increase the reactive species, which causes increase in lipid peroxidation, DNA damage, and also protein oxidation. These reactive species are able to impair several pathways or activities inside the cell, which will impair the GLUT4 translocation to the membrane, consequently impairing the diabetic condition and also related to the depression-like behavior related to this diabetic condition. So this is a very interesting overview and focus, and we can observe that inflammatory cytokines seems to be a very important role in this process. This study is very interesting because they show that hyperglycemia driving neuroinflammation compromise blood-brain barrier. So there is no surprise that this occurs, this impairment on blood-brain barrier, increasing even more the inflammation condition in the central nervous system. Well, just to reinforce the role of inflammation depression associated to diabetes, in this study, the authors indicated how important are the associations between inflammation-related biomarkers and depressive symptoms in individuals with recently diagnosed type 1 and type 2 diabetes. So this is a very interesting discussing, highlighting the involvement of inflammation in this process, reinforcing our hypothesis that neuroinflammation also could be a key factor relating depression and diabetes. As a partial conclusion, we could infer that diabetes mellitus is a condition able to increase pro-inflammatory cytokines in some brain areas like hippocampus. We did not show data involving oxidative stress, but we have some finds in our lab, and several other research confirm this data, that there is also an increase in oxidative stress in the hippocampus and prefrontal cortex. And as we demonstrated, other groups also demonstrate a decrease in serotonin levels in some brain areas like hippocampus, and all these changes seems to be involved with the depression. And we and Bambi and co-works also showed that there is an increase in the idle activity or expression, probably relating with this decrease in serotonin, and also with this, probably these pro-inflammatory conditions are able to increase the activity of idle and even the expression, and this will be related to the decrease of serotonin synthesis or release. And we the animals with neuroprotective compounds like fluoxetine, minocycline, ibuprofen. These treatment were able to decrease the depression, depressive-like behaviors, and in these animals, and we could infer probably these treatments also serotonin levels. Recently, we demonstrated that a prolonged treatment with the specialized pro-resolving lipid mediator resolvin D5, which is an orchestrator of the resolution of inflammation, was able to prevent the increase in brain levels of interleukin-1 beta. We observed this in the hippocampus and in the prefrontal cortex. And this reduction in this pro-inflammatory cytokine was correlated with some improvement on the depressive-like behavior and anxiety-like behavior in these same diabetic animals. Interestingly, this prolonged treatment also improved some parameters related to diabetic condition. So here we can observe a decrease in the hyperglycemia, especially with the highest dose, and also an improvement in the weight gain of these diabetic animals. To conclude, all these changes seems to be interconnected. Inflammation, oxidative stress, decrease in monoamides like serotonin, and we did not mention, but also this is observed in this in the brain of diabetic animals, an increase in glutamate activity and decrease in neurogenesis, and also in the synthesis or action of BDNF. So the neuroinflammation seems to be a key factor in depression associated with diabetes mellitus. So, and it seems to be a very important target, because when we have this increase, we will have several other changes leading to the several complications or comorbidities related to the diabetes. Like to thank all the researchers involved, my students, and also some colleagues from my department, and also from the Aarhus University, Estadual University of Londrina, and also from University of São Paulo. And thank you for your attention. Okay, thank you for your attention too. Gisele asked me something about GLP-1 analogs. Yeah, there are some studies involving this compound, examgene 4, for example. And what I know is that the evidence is a little confused because it depends if it's acute treatment or chronic treatment. And there are some study in which they investigate, for example, anxiety-like response and depression-like response in rats treated acutely or chronically with some analogs, and they show different effects. For example, anxiogenic-like effects and changing in serotonin levels in some brain areas. They investigated the amygdala in these animals. But if you perform a more prolonged treatment, like chronic treatment, you could observe no change in anxiety-like response, but antidepressant-like effects. So it's very interesting because we know the effects of this compound improving several intracellular pathways, inhibiting inflammation also. So, but I didn't evaluate. I am just I have the interest in studying a little more about these compounds in our modules, but they are studying also in animal model of type 2 diabetes, which is more interesting in some aspects, you know. But well, that's it. If what the difference conclusion between prefrontal and hippocampus experiment that can we learn? it's it's a very curious question because our lab and several authors have been focused in hippocampus and prefrontal cortex. What we have seen is some difference, for example, in neurotransmitter levels. In prefrontal cortex, for example, there is an increase in noradrenaline, while on hippocampus is a decrease. So there are differences, of course, but in several other parameters, they look like similar in changes, at least in pro-inflammatory cytokines, IDO, but I don't know the difference between inflammation markers or neurotransmitter levels, how the impact of this difference is in the behavior of the animals. But of course, there are some evidence showing difference between these two brain areas. We know that both brain areas are important to the mediation of depression, learning, memory, and also insight. So we need to study and focus about maybe some specific brain areas like infralimbic. We don't have so many studies, especially in my area like diabetes and depression and anxiety, focus on different parts of medial prefrontal cortex, for example. And we know there are difference between these infralimbic, prelimbic areas. Probably this data showing same effect is because we need to focus more specifically in this within the medial prefrontal cortex. No? I don't know if I answered. It's a little complicated. We needed to focus better in these details, in these specific pathways, because these data I have shown you is a kind of a first data. So now we needed to perform more detailed experiments. So we need grants to do this in our lab. Sometimes we have this difficulty here, like to buy reagents, drugs, antibodies, and but sometimes we have very nice collaboration and we can do something more in detail and focus more on some pathways, for example. Thank you. So, well, thanks a lot for all my my speakers. I loved all the presentation. I think our idea about inflammation now was highlighted by all of our speakers. And thanks for who stay here until now. I know you are hungry, so thanks a lot. Thanks, Beth. So we can close this session now and have a very, very nice afternoon for all of you. Bye. Thanks, Ibrahim. Obrigada, Ibrahim, por sua ajuda.
Janaína, Gisele, Josiane.
Eu que agradeço. Nos vemos durante o evento.
Thank you.
Obrigada. Abraço a todos.
Obrigada. Abraço.
Vamos nos falando.
Tchau, tchau, gente. Obrigada.